Okay. So today we are talking about antibodies. These are circular proteins that are produced in response to exposure to a foreign structure. So these are part of the adaptive immune system. Last week we talked about the innate immune system that's ready to go. It is produced outside of a response to foreign structures, but this is adaptive. So um, these are produced after an antigen has been seen by the immune system. Since they are adaptive, they are highly diverse and extraordinarily specific, again, in contrast to the innate immune system. Um, I added this because this is on the Fit Bowl from time to time, but they were first identified by von Behring and Kitasato in 1890. Um, they were recognized to inactivate toxins um, and induce immunity. It was also noted they could be transferred to other animals, although this was largely known before, or at least the concept of it, but structurally they were not identified before. They were called the humors, which could be transferred from one animal to another after immunity was induced in one animal or human, but that stuck. So the humoral immune system is based on these humors or antibodies. Um, and an example of an early use was diphtheria was treated with an immunized horse serum. So they gave the toxin to the horse, it induced immunity, and then that could be transferred actually to another animal or human to treat the disease. Okay, so antibodies can be found in two forms. One is a membrane-bound form, which I personally don't think of as antibody much, but they are. But we also call this a B cell receptor. Um, and they can be in the secreted form, which clinically is what we deal with a bit more. They have lots of functions. I think the most obvious is they neutralize antigens. So they neutralize microbes, toxins, but they will also activate the complement system and they also will opsonize. They participate in um, antibody dependent cytotoxicity, and they will also activate mast cells to release their preformed mediators to treat parasitic infections or helminthic infections. And of course, the pathology involved in that is immediate hypersensitivity. So in the upper left here, you see the structure of an antibody or an immunoglobulin. And it has some redundancy with other receptors that we're going to see throughout this book in most of the chapters from here on out. Um, there is an antigen binding site here that is made up of these complementary determining regions. It has two variable variable domains and then a constant domain down here. So here's where all the variability comes in the antibody in the top right and left here. And this is where the antigen is recognized. And then the actual function is described in the structure here. We have two heavy chains and then two light chains. Structure is similar in this T cell receptor. You can see there are CDRs and there's kind of the symmetry in the chain similar to MHC molecules as well. Okay, so they're found in biologic fluids throughout the body and on the surface of B cells. They're initially produced in lymphoid tissue in response to interaction with the T cell. But then once B cells become plasma cells, they move to the bone marrow where they reside and then secrete antibody from there. Okay, so in general, serum proteins are separated into albumins and globulins. We can separate these by electrophoresis, and antibodies are in the third fastest migrating group of globulins. alpha, beta, gamma. So they are called gamma globulins. So you may hear when we're talking about IgG replacement gamma globulins, or you could hear that just as a synonym for antibody um, or immunoglobulins. Some people just call them gamma globulins. So that's probably not important to know outside of really just understanding where this term came from and that it really essentially means the same thing as antibodies. Okay, so breaking down a little bit again, here is on the left a secreted antibody. This particular one is an IgG, and on the right there is a membrane-bound IgM. They're extremely similar, except for this constant region down at the bottom that goes through the plasma membrane of B cells. Um, so both of these, you can see these, the way Abbas draws these are kind of these C or U shapes, and these are immunoglobulin domains. This particular motif, so if you have lots of amino acids, they fold in a very specific manner that is highly redundant across many different receptors of the immune system. So you're going to see them drawn in these pictures the same way throughout, and it just is trying to demonstrate how similar or redundant these receptors are to each other. Um, okay, so here you can see you have these um, you have these variable regions or immunoglobulin domains here at the tip top, and then you have a series on the light chain of just one, but on the heavy chain, three more constant regions um, times two. So this is where the antibody is going to bind, and this is called the FC region. This is where the effector function is going to be. Same on the membrane, except of course, same on the membrane bound form, but this is actually going to then start a signal. Its effector function is to start a signal like other receptors through the cell. Okay, so here's this immunoglobulin binding domain, these amino acids that have folded into a similar region, and this is just looking at other receptors that we're going to talk about throughout the course of this book and kind of that homology that exists. You can see even in a T cell receptor, similar to a B cell receptor, you have these variable and constant immunoglobulin domains, but it's also even preserved through MHC, which we'll continue to talk about. Sorry, there's construction going on in my building. I know it's distracting. Okay, so in these variable domains, there are some loops that come out, and they're called complementary determining regions, and there's three of them. We call them CDR1, CDR2, and CDR3. So CDR3 is the most diverse. That has the most diversity to it. And this is where the antigen binds to. So here on the outside where the antigen is binding, you have these immunoglobulin, but then you're going to have these CDR pieces that stick out. And I think in the next picture here, you can see this well. So you have all these amino acids lined up, and they actually fold into these beta pleated sheets, and it kind of makes this nice pattern. But then you get these little pieces that stick out. This is the CDR. This is where there's a lot of variability. They call it the hypervariability. And CDR3 is the one that is the most diverse. This is just one more picture to help drive that concept home. Here you have your heavy chain. Of course, this is going to continue on. And then you have your light chains. And then you have these CDRs, and this is where the antigen is going to bind. This actually becomes the antigen binding site. So here is both an antibody binding to two different antigens. You see this Y pattern, and then you see all these immunoglobulin domains with the beta pleated sheet. So it's all folded in this characteristic way. And then you have the CDR. This is what's going to bind to the antigen. There's also a specialized region called the hinge that is just on the heavy chains. And what this allows it to do is for the antibody to bind more easily to different antigens. So here's just an example where on the outside of a membrane, it's recognizing an antigen that's sort of widely spaced, but it also could be narrowly spaced. And there's a hinge there that allows for some flexibility for how close those antigens are for this still to be effective in activation. Um, this is, just another one of those kind of random facts. I really don't know if you're not a basic scientist, we don't use this clinically that it has that much relevance, but there are two different um, chemicals that you can use to degrade antibodies. One is called papain, and one is called pepsin. Pepsin's here on the right, and papain's on the left. So papain, when you use it to degrade an antibody, it selectively targets the region above the hinge. So then when papain degrades the antibody, then you get two FAB regions and then one FC regions. So two where you have this antibody binding. But if you use pepsin, it actually degrades it below the hinge. So when it binds it below the hinge, all you get is one FAB2, and then you get these peptide fragments of what the other part of the heavy chain would be. So it's a little bit of difference. I don't know if it's still necessary to know this or not. I actually I can remember memorizing that for boards, but I honestly don't remember if it comes up or not. Oh, I always put, actually, I forgot. I have a mnemonic for this one that's very goofy. But papain is derived from papaya. So I always remember this, that papain comes from papayas, which are fabulous. And you get these two FAB regions. So that's my mnemonic to remember papain and pepsin. Okay, so the light chain, each antibody has one or the other. About 60% of the light chains in your body are kappa and 40% are gamma light chains. Each antibody has different, or two antibodies that have different isotopes, but we have, therefore, a diverse set of different antibodies. So IgA can actually have IgA1 or IgA2. Then you have IgM, IgG, and IgG has four types: IgG1, IgG2, IgG3, and IgG4. Of course, the antibody that clinically we check less, but not, just in periodic fever syndromes, we check IgD, but we actually don't have just on an antibody panel. But these are all the different antibody isotypes that there are. And each isotype has a slightly different function and secreted form. And this is, I think, a very important point. So there are two antibodies in the secreted form that are monomers. So this is IgE and IgG. The rest, so IgD is not in a secreted form, it's just on B cells. IgA in the secreted form is always a dimer. So there is a J chain here that connects two IgAs to form this one IgA dimer. And its function is in mucosal immunity. So this is largely secreted in different secretions in the body, like saliva, for example. IgE, of course, is important in this defense against helminthic parasites. IgG has highly diverse functions, including opsonization, complement activation, the antibody dependent cell-mediated cytotoxicity, neonatal immunity. This is, of course, what is passed to baby from mom over the placenta, and feedback inhibition of B cells. IgM is by far the largest secreted antibody because it's found in primarily pentamers. So 5 different antibodies that are all connected by a J chain. IgM is very important for complement activation. Okay, when antibodies are synthesized, they're synthesized on ribosomes, as proteins are in the endoplasmic reticulum, and their folding is regulated by chaperone molecules, including calnexin and BIP. And then they go to the Golgi apparatus for modification. Then they're transported in membranes to the membranes in vesicles. So we can actually look at B cells and the different stages of maturation based on how formed the antibody associated with it is. Okay, so what starts as a stem cell will become a pre-B cell. A pre-B cell is made up of a mu heavy chain and a pre-B cell receptor. And then you get an immature B cell where you have a membrane IgM, but then a mature B cell has IgM and IgD on the outside. Once it's activated, it becomes a plasma cell and actually secretes antibody. So I bring up the mu heavy chain specifically because there is a identified mutation in mu heavy chain in patients with a GAM that is very similar to Bruton's tyrosine kinase, which is, if you're on the PEDS board, you're probably going to be tested about Bruton's. But on the allergy board, you very well may get what seems like it should be Bruton's, but it is actually in a girl. And they're talking about this autosomal recessive form, which is a mutation in the mu heavy chain. Okay, so each of these isotopes have different half-lives. So there's IgG, which has a very short half-life at 2 days. IgA, also very short, 3 days. IgM, just 4 days. But the one that is different and important to remember is IgG. So this is 3 to 4 weeks. And the reason this is important to remember in my mind is because clinically we always dose medications once every half-life. And we know medications wash out every 5 to 6 half-lives. So if you think of two different circumstances, one when a baby is born, 5 to 6 half-lives, of course, 5 to 6 months, they should have washed out completely of mom's IgG. In those first few months, they shouldn't be making more IgG. So if you check an IgG 1 week and you check it again the next week and it's higher, well, that may mean that they're making their own IgG, or probably means they're making their own IgG. But it should be degrading one half-life. So every month, by the time they get to 5 to 6 half-lives, it should be completely washed out. The other is when we're talking about IVIG, if we're giving immunoglobulin replacement in an IV version, we dose it once every half-life. So once a month, they get an IgG infusion. And then if you're waiting it to wash out because you want to reevaluate their immune system, you have to wait a full 5 to 6 half-lives, 5 to 6 months before you really can be sure that what you're testing in the immune system is actually your patients, and it is not what was donated in the IgG IV that they received. There's another interesting point here that there is something called the neonatal FC receptor. And what this receptor does is it transports IgG from maternal circulation across the placenta, which of course is very important for that first point I was talking about. And it will transport IgG across the neonatal intestine. And it also sequesters IgG intercellularly. Another fact is that in recent years, it has served as a way to extend the half-life of certain pharmaceuticals, namely biologic therapy. So if you have a monoclonal antibody or an IgG specific to something like an IgG specific to IgE, in the case of omalizumab, one way that they will try and increase the half-life of these biologics is by combining that IgG to this neonatal FC receptor. It retains it in the circulation longer by doing so. Okay. So antibodies can recognize several different things. So it can recognize metabolites, sugars, lipids, autocoids, hormones, phospholipids, nucleic acid, but it can most strongly recognize proteins. And proteins, when I think about it, is clinically one of the most important, particularly when we are working up immune deficiency. We look at how it is acting against proteins. But of course, we go back and we also try and figure out how it's working against polysaccharides as well. So antigen binding, is important for B cell activation. So when it's a membrane antibody, there is a redundancy in the immune system, which we've talked about before, where you really want this checks and balances, and you don't want the immune system to have one signal inactivate because it could be aberrant. You want multiple signals kind of as a check and balance before you really activate the immune system. So for B cell, this can happen in two ways. Either one, and again, the most common where you have these proteins, it requires T cell help. So you get that in the lymph node, that T cell, which recognizes a protein, and then a B cell, which gets activated by recognition of that same protein in interaction with that T cell. But there's also scenarios where you can actually use cross-linking. So polysaccharides have the antigen repeats. And if you activate several antibody binding sites at the same time, that can also, in a less powerful way, activate B cells as well. There is also a way to improve antibody response, and that is to use something called a hapten. And a hapten is when you actually put an extra portion on an antigen to improve the likelihood that it's actually going to get recognized by a B cell. Haptens can be exploited when we are trying to make vaccines. So these are all things that we think about when we are working up immune deficiency, but these are also things that we think about in vaccine development, about how we actually get the immune system to respond to the antigen that we are deciding goes into the body. So the part that the antibody will actually recognize is a determinant or an epitope. And if there are multiple identical epitopes on one antigen, this is called multivalency. So again, we get to that redundancy, the importance of that redundancy. So epitopes can be linear or conformational. I think clinically we see this in food allergy all the time. So linear determinants are about 6 amino acid long, and it's just how it says, it's 6 amino acids in a row. But then there are conformational determinants, and these are made only after the protein is folded, and it actually recognizes different pieces and different lines. So as soon as a protein is denatured, that determinant will go away. So here, I think, is really what we want to see. Here's very simply a linear determinant. So here's your protein that's getting recognized. And along the line here, you may have about 6 amino acids that's recognized by this antibody. Sorry. Can you hear me at all through this? Or did you say you can't hear the reflection?
No.
Oh, good. Okay. Then I can continue on. That's all I can hear. Okay. So linear epitope, if it's denatured, it does not change. That linear epitope will stand. So we think in like a more severe egg allergy or one that is more likely. It doesn't matter if you denature or bake egg, same with milk, then it does not change the reactivity. You're still going to recognize it. But many epitopes are conformational. So it depends on how it's folded, and as soon as it's denatured, it goes away. So these are tends to be less severe forms of milk and egg allergy that we know that we can actually exploit that denaturation and get some of these foods into babies. Okay, so when antibody binds to an antigen, it's non-covalent and reversible. It has different affinities, which are marked as KD. So a smaller KD means it has a stronger affinity. But again, it is a non-covalent reversible bind. There is a avidity, and this speaks to the overall strength of the attachment and is actually increased. I think I have a nice picture here by the number of sites you have to actually bind to the antigen. So here is monovalent. This would be something like an IgG binding in just one side. It has a very low avidity. Bivalent is where you have both of the antibody regions of the antibody binding. But then when you have a secreted IgM, remember, which comes in a pentamer bind with a J chain, then you actually have 10 different opportunities for antibody to be bound. Is we call polyvalent. It has a very high avidity, not affinity. So try not to mix those two terms up. There's also a concept of the zone of antibody access, where you have lots of antibody and a small number of complexes. And then you have a zone of antigen access, where the antigen actually outnumbers the number of antigen binding regions. But then there could be a zone of equivalence where they're actually perfectly matched, where you could, in a pathologic way, develop large complexes that then can deposit in the body, like in the joints, for example. Okay. So remember, these are very specific, they're very diverse, and they undergo something called affinity maturation. So they are very specific. Remember, this is part of the adaptive immune system. So we actually get DNA rearrangement. So we have lots of diversity. In fact, it's estimated to be about 10 to the 11th. And this is called, all that diversity is called your antibody repertoire, to talk about all the different types of antigens that could be responded to by the different antibodies you have. Of course, we exploit this by taking 1,000 different donors who will all donate antibody, and then we'll make it into a formulation of IgG replacement. Each individual who donates will have their own diverse antibody repertoire, and the thought is if you're combining thousands, then we're really getting a very broad range. You're getting all these different antibody repertoires to cover one person and their immune system if they have an immune deficiency. There is also something that is called affinity maturation, which can increase the specificity or increase that binding affinity. So the likelihood that any antigen will actually bind to the antibody. This happens by somatic hypermutations. I actually forgot. Okay, maybe we'll talk about that in a second. Okay, so the effector function, which I mentioned briefly before, is dictated by the FC region. So this is different based on the different isotope. And it's only effective when bound. So there is a phenomenon called isotype switching, where initially IgM, of course, membrane-bound IgM recognizes the antibody, and then it's secreted IgM, and then you get isotope switching. So then you can actually switch to an IgA or an IgG. This will also determine the tissue distribution. Okay. So once again, here's the different parts of your antibody. Here you have the variable region that's going to bind to the antigen that you can see in yellow. And then you have this constant region. Here's the membrane bound. So you have the transmembrane with a cytoplasmic tail that is going to communicate with membranes to send a signal. So this original IgM, this membrane-bound IgM, can undergo something called affinity maturation. And this is where, and we'll talk about this more in other chapters, but this particular antibody structure will undergo a series of mutations. And then it's tested against the antigen to see which has increased affinity, not avidity here, increased affinity. And when it has increased affinity, it will be selected. The ones that have decreased affinity because of these mutations will then be selected out. So they will be killed out. Once you have undergone this affinity maturation, it will switch to a membrane, from membrane to a secreted form. where it does not change anymore. And then finally, it can undergo isotype switching, so it can actually change the function of the antibody.